Jackson is coming to us from Beijing, but he was born in New York City and got a did an undergrad degree in physics and math here. And actually, sorry, before that, went to LCC. What did you, I want to say you did high school at LCC, but what do I say you did at LCC?
Science.
Science at LCC. Okay. Then got a master's in physics from UCLA, switched to biology, working on microbiology and immunology, getting a PhD at City Hope Beckman Research Institute, was a postdoc at Cornell with Philip Messer, and opened a lab at Peking University in May 2021. Since then, he's been an assistant professor in the Center for Bioinformatics at the School of Life Sciences and the Center for Life Sciences. And he works on things that will be unsurprising given his title. And afterwards, we'll have tea time up in that one room where it always is, 318 Pacific. Everybody's welcome to come. And if anybody wants to talk to Jackson this afternoon, there's some spots, or go get pizza, talk to me. Thank you.
Thanks. Yeah, thanks for inviting me. So besides doing college here, this is still where a lot of my family members live. So very convenient to be visiting right now. It's actually the Spring Festival holiday in China, so much easier for me to escape at this time. All right, so my lab is all about gene drive and closely related areas. So first of all, what is a gene drive? There are both natural and engineered versions of gene drive, but my lab will specialize in the engineered versions. These are genetic packages that you can insert into organisms. So then you've got these transgenic gene drive organisms. You release them into a wild population, and then over time, the gene drive allele will spread through the entire population. So this is a potentially very powerful way to manipulate entire populations in the wild with just a very small investment of resources, just a small number of initial gene drive individuals. Here's how it works. On the left there, we have a normal allele in a heterozygote, the one in orange there. By Mendelian inheritance, that allele will be passed on to half of its offspring in each generation. That means, as you see on the bottom, if there's no expected fitness costs, the allele on average will remain at the same frequency in the population. A gene drive allele, though, is able to bias its inheritance. So that's red in the top right. You see that that red gene drive allele, even though it's a heterozygote, is actually passed on to all of the offspring in each generation. It's a perfectly efficient gene drive, and that means on the bottom there, in red, you see that in just a few generations, it reaches the entire population after only a 5% release. There's two basic things we can do with the gene drive. The first is population modification. Basically, we spread the gene drive through the whole population, and it's also carrying some desired trait, such as a gene that prevents mosquitoes from transmitting disease. You can also configure a gene drive to do population suppression. We'll talk a little bit more about how that works later on. Of course, as you see from the mosquito pictures here, I think that those are the most interesting applications. Right now, malaria will kill 500,000 people every single year, most of which are children. It's spread by Anopheles gambiae and other Anopheles mosquitoes, but there are already cargo chains that can effectively prevent transmission of malaria if they're in the mosquitoes. We just need a way to get it to all of them. There are many other mosquito-borne diseases. Those 80 mosquitoes, for example, will spread a lot of dengue and other viral diseases. And even these Culex mosquitoes can carry a few nasty ones. For some, though not all of these diseases, we also have existing cargo genes that can prevent their transmission. But we can do a lot more with gene drives. All over the world, there are invasive species which are causing huge damage to ecosystems. The alligator gar is a particularly nasty example in China, redwood pigweed all over Asia. The cane toad in Australia is one of the most classic examples. It was brought in to control a pest, but ended up becoming one of the biggest pests of all. It eats other amphibians, lizards, crops, anything it can get its hands on. Rats might be a cute and important part of many ecosystems, but when they get to tropical islands or even bigger islands like New Zealand, they'll start eating all the birds and other life forms and have already caused many extinctions. And then besides for natural ecosystems, we also have agricultural lands. Represented by the spotted wing Drosophila, these agricultural pests do huge damage, and the pesticides used to control them can also have harmful environmental effects. A gene drive configured for suppression could potentially address all of these problems if we can suppress these species where they're causing these difficulties. So that's what we can do with the gene drive. How does it actually work at the molecular level? Well, there are actually multiple different types of gene drives. We'll get into that in a lot more detail later on in the talk. But right now, I'm just going to tell you about the homing drive, which is the most commonly studied at least, and the most powerful type of gene drive. It works by using a nuclease, usually CRISPR-Cas9, to cut the wild type allele on the chromosome opposite the gene drive allele. Then we've got a double-stranded DNA break. Anytime that happens, the cell machinery is going to need to fix that break, otherwise the whole cell will die. But cells are really good at that. In this case, you can see that the DNA on either side of the break is identical to the DNA on either side of the gene drive allele. That means that the cell can use the gene drive allele as a template for homology-directed repair, and that will copy the gene drive allele into the wild-type chromosome during this process of repair. If this happens in the germline reproductive cells, then you can see that even though most of the cells in the organism are heterozygous, those germline reproductive cells are homozygous. And that is how over half of the offspring can inherit the gene drive. Unfortunately, homology-directed repair is only one possible repair pathway. There is also end-joining repair. And in end-joining, we won't use the gene drive allele as a template. Instead, we'll stitch the ends back together with a complicated molecular mechanism that often produces an indel mutation. We call that a resistance allele because it can no longer be recognized and cleaved by the drive's guide RNA, which requires a specific sequence. This changed sequence, thus, can never be converted to a drive allele. And these resistance alleles are one of the biggest challenges we face in making effective gene drives. So why is that a problem? Well, even if we have an extremely efficient drive, here it reaches 99% of the population there in red. You see that at the bottom, a very small number of resistance alleles has formed by the time the drive reached its highest level. This resistance allele, though, has a small fitness advantage over the gene drive. And that means over time, it will outcompete the gene drive and eventually take over the population. The gene drive will fail. Really, though, there are two types of resistance alleles. And only one of them is a big problem. Most gene drives will have a target gene. And it's the functional resistance alleles that will have a very high fitness. These alleles have just very small mutations. Maybe it's a silent mutation, or maybe the amino acids are mostly similar to the original amino acids of the protein. So the protein still works. And that's why it will have a fitness advantage over the gene drive. Most resistance alleles, though, will have a disrupted non-functional sequence. 2/3 of the time, we'll get a frame shift mutation. And most of the rest of the time, we'll change the amino acids enough so that the protein won't work anymore. And we tend to get quite a lot of non-functional resistance allele formation. But it only takes a small number of functional resistance alleles to ruin the gene drive. And in fact, for a suppression drive, well, this would happen even faster. The resistance would actually increase almost as quickly as the gene drive did. So how can we address that? The key is gRNA multiplexing. If instead of using one guide RNA, we use several guide RNAs targeting close together, then as you can see on the left there, each one will need to be repaired perfectly for the entire allele to be functional. If even one of them is repaired imperfectly, as you can see in several of these examples on the right, well, then the whole thing just won't work. So this means that with gRNA multiplexing, we can make it exponentially less likely to get functional resistance alleles. And that can solve one of the biggest problems we're having with these. Besides that, it's actually even better. If we cut out an entire piece of the wild type allele, well, then it will definitely be non-functional. So with just a few guide RNAs, we can prevent functional resistance alleles from causing problems. So how can we use that to make a good modification gene drive? Here's an example of one we actually made in the lab. It targets the haplolethal gene RPL35A, very important for ribosomal proteins. The organism needs 2 copies of this essential gene to survive. We're targeting this gene with 2 guide RNAs, which in the lab should mean that all the resistance alleles are non-functional, and then they're removed. So what else does this drive have in it? Well, besides the guide RNAs, it has a DS-red fluorescent protein. That will make all of the insects glow red in their eyes, allowing us to very easily track which ones have the gene drive. We've also got in purple there a recoded right sequence. That's basically a rescue element for this gene drive. Normally, the gene drive itself would go right into the middle of the target gene, which would definitely break it. But with this rescue element, it completes the right half of the target gene. And that means the gene drive will always have a working copy of RPL35A. We've changed the nucleotide sequences, though, but still keeping the same amino acids so that the guide RNAs of the gene drive won't cut itself. Another thing you might notice about this gene drive is that we put the Cas9 at a different genomic site. Usually it would go in the middle of the gene drive. But if we release these flies out into the wild, well, even if it's an accident, it might just take one fly to spread this in the wild population. That would get us in big trouble, and we didn't want that to happen. So instead, we took Cas9 out of the gene drive, moved it to a different genomic site, and just provided it in all of our experiments in the lab. This way, in the lab, it would act like a normal gene drive, but if these were released in the wild, very quickly the Cas9 would be eliminated because it can't copy itself. Only the drive element can, and that means it won't be able to spread in the wild. It'll just stick around for a few generations before being eliminated due to fitness costs. So let's go over how this works. In the germline, we prefer to do drive conversion, converting that blue wild type allele into a red gene drive allele. But sometimes we'll make non-functional resistance alleles. Then meiosis happens and fertilization. And as you can see, after fertilization, everyone inheriting even one resistance allele is immediately removed from the population. But there's actually a problem. You see, in that embryo Cas9 activity down there, we can make additional resistance alleles. If the mother has the gene drive, she'll make so much Cas9 and guide RNA in the germline that there's still a lot left over in the new eggs. And anytime we have Cas9 and guide RNA, we can cut wild type alleles and turn them into resistance alleles. In the one on the left, you might think, oh, wait a minute, there's a drive allele there. So if we cut it, can't we just do drive conversion and turn it into a drive allele? Well, that's actually a very common point of confusion. And even in the first CRISPR gene drive paper, people were confused about that. But as it turns out, that embryo development is so fast, there's probably just not enough time for homology-directed repair. We've only found resistance allele formation in those early embryos. On the right there, it's just getting rid of another wild type allele. But on the left there, you can see it's making the gene drive non-viable. The gene drive might have a good copy, but it's only got one of them. So that gene drive is removed from the population, which will, of course, hurt the gene drive quite a lot. Fortunately, in this particular drive, that only happened about 30% of the time. The gene drive was still efficient enough to work well. When we released this gene drive into a cage population, slightly higher frequency than we intended, but ultimately it didn't affect the population dynamics, it increased very quickly and soon reached 100% in a cage of a few thousand flies. This was the first successful homing modification drive. So that's how we can make a modification drive that will work. What about population suppression? Here's a computational model of population suppression success as a function of the number of guide RNAs. We do make a lot of functional resistance alleles in this model, even though it's a very efficient drive. So you can see that if we have a very small number of guide RNAs, we almost always fail to eliminate the target population. If we have an intermediate number, then it works pretty well. But when we have a lot of guide RNAs, we start failing again. So why is that? As it turns out, there are limitations to the multiple guide RNA strategy. If we add too many guide RNAs, beyond 2, in fact, the efficiency of the gene drive in terms of converting wild type alleles into gene drive alleles will actually go down. And suppression gene drives are very sensitive to the efficiency of the drive. Here's why. A suppression drive targets an essential but haplosufficient gene, usually a female fertility gene, so that we have more drive power. So what does that mean? So haplosufficient, haplo means one, sufficient means enough. So one good copy of this target gene is enough for these females to be fertile. The gene drive doesn't provide a rescue element here. It goes right into the middle of the gene and breaks it. But as you can see in the left here, these female drive wild type heterozygotes are still fully fertile. They can do drive conversion in the germline, so they can still spread the gene drive. And then, as the gene drive reaches a high frequency, we'll get more and more female homozygotes. Those ones will be sterile. So the idea is that the gene drive spreads to a high level. We get mostly sterile females, and then the population collapses. Unfortunately, when we tried to do this experiment with a 4 guide RNA drive, we failed to eliminate the population. As you can see in these 2 cages, it increased in frequency at first, but then it kind of pooped out around 60 or 70% carrier frequency. There were definitely a lot of sterile females here, but not enough to affect the population of our robust fruit flies. Why was that? Well, an imperfect suppression drive will actually create a balance between drive conversion in heterozygotes, which increases drive allele frequency, and removal of drive alleles in female homozygotes. If the drive is perfect, the upward force of the drive will always be greater. So it will still increase to 100% and always eliminate the population. But if we can't convert all the wild type alleles into drive alleles, then eventually we'll reach an equilibrium. And at that equilibrium, well, maybe we'll have enough sterile females to eliminate the population, maybe not. It depends on exactly how strong the drive is. We can also run into other problems with non-functional resistance alleles. They won't completely stop and eliminate the drive, but they can reduce its suppressive power, especially if there's more of that embryo resistance due to maternally deposited Cas9. That might make sterile females, but it often makes them too soon and gets rid of drive alleles that we needed to increase the population. In these cages, we got too much of that maternal deposition, and that is why the drive was a failure. Fortunately, we can make upgrades. By using a different Cas9 promoter, instead of our nanos promoter, which has a lot of maternal deposition, we found these two other promoters, CG4415 and RCD1R, that have much less maternal deposition. So much less resistance allele formation. Unfortunately, RCD1R still didn't work. It might have had less maternal deposition, but it also had somatic expression, which is another problem that suppression drives have. If we get rid of that wild type allele in the female heterozygotes, then the females will be much less fertile. So RCD1R reach an even lower level than nanos. But for CG4415, for both this replicate and several others, the drive reached a high frequency and was eventually able to eliminate the population. So by making a higher performance suppression drive, we eventually were able to achieve success in these model organisms. So our next step was to make a better gene drive in mosquitoes. Other groups have already been working on suppression drives in Anopheles gambiae, the biggest malaria mosquito in Africa. But Anopheles stevensi is a growing problem as well. It was originally found in South Asia, where it still causes a lot of malaria, but it's now also invasive in both East and West Africa. The tricky thing about stevensi is that it's a city mosquito. It loves urban environments, and with increasing urbanization in places like Africa and India, a lot of people think that Anopheles stevensi might start to set back a lot of the progress we've made against malaria over the past couple decades. So it would be quite nice if we can address this mosquito as well. We made several upgrades to the original suppression drive target in Anopheles gambiae that targeted double sex. It goes after the female-specific exon, so that's just like going after a female fertility gene. But the nice thing about this target site is that it's super, super conserved. Functional resistance alleles would be extremely rare, even with one guide RNA. But we knew that still wouldn't be enough to prevent functional resistance in mosquito populations of hundreds of millions compared to the original laboratory population of just a few hundred. So we added a second guide RNA. We also used the nanos promoter here, which in Anopheles gambiae was a great promoter that had low fitness costs and low maternal deposition, even better than the original promoter that they used. Unfortunately, this promoter was not so great for Anopheles stevensi. You can see here that only about 3/4 of progeny from drive heterozygotes inherited the drive allele. That means that this drive won't be powerful enough to actually eliminate real Anopheles stevensi populations. We'll need to try a different promoter for Cas9 to get higher cut rates. And in fact, that's a really big problem in general for population suppression drives. There's a lot of applications where we might need to do population suppression, but the requirements for a good population suppression drive in many different species are very harsh. Here's a computational analysis of a normal female fertility suppression drive. The genetic load here shows the suppressive power. You can sort of think of it as how many females are sterile when the drive reaches its equilibrium. You can see that the drive conversion rate, the drive efficiency, needs to be very high to get high genetic load. If we have a fixed drive conversion rate and increase the total cut rate, that just makes more non-functional resistance alleles in the germline. These resistance alleles don't hurt us that much, but you can see they don't do anything to actually improve the genetic load of these drives. So if we can't make an almost perfect drive, we won't have enough suppressive power. And we've only made drives that are good enough in Anopheles gambiae and Drosophila melanogaster. For almost all other organisms, such as mice, Aedes aegypti, other agricultural pests, we're not going to have very good drive conversion efficiency unless we get very lucky with the gene drive. So we need a new design for a suppression drive. Here's the design. It combines the homing modification drive there on top, just like the one I showed you earlier, with additional guide RNAs that target the female fertility gene there at the bottom. So the drive doesn't directly copy itself into the female fertility gene. It just cuts it and then does either HDR or engineering repair to disrupt it. Since the drive is no longer inside the female fertility gene, it won't suffer a lot of the fitness costs that it did previously. So it can reach a much higher level. Here's a graph of the genetic load, the suppressive power of this new design. You can see that even with low drive conversion efficiency, as long as we have high total cut rates, we'll still have exceptionally high genetic load. And it has been much easier to get high total cut rate than high drive efficiency. So we believe that this new design might be very useful in a lot of non-model organisms in the future, allowing us to bring gene drive to more difficult species. And just as a little fun slide, back in 2009 to 2010, just before I started my PhD, I was a research technician in the Kimmel lab, making fluorescent zebrafish, many, many different colors. And quite recently, at Peking University, we started a fish drive collaboration. We've got fish that have fewer fluorescent colors, as you can see here on the right, but there might be a very interesting meaning behind that fluorescence. Perhaps we've made a decent gene drive in these fish. There are all sorts of invasive fish in the world, so hopefully these are the first steps to getting rid of those fish where they're not supposed to be. All right, change of topics. We're divided into 4 different topics here. So if you fall asleep in one of these topics, it should be completely fine, as long as you're awake by the start of the next topic. So this next topic will be about computational modeling of suppression drives. Most computational models have very simple populations. All the individuals compete together. Any individual can mate with any other. Some people on the left there will have 2 of these populations linked by a fixed amount of migration. But for many species, at least, these might not be the best models. Real-world populations are spread over landscapes, continuous landscapes with forests, mountains, rivers, streams. We're not quite at that level yet. But at the very least, we can make a model that has continuous space. Each of these blue dots is a wild-type organism. It can only find mates nearby. It only competes nearby. It only disperses offspring nearby. So let's see what happens if we release a suppression gene drive in red into the middle of this population. The gene drive will spread through the population. Pretty soon, it's eliminated. It took a little bit longer than in a simple model because the suppression drive needed to spread from the center to those corners, but it was still pretty fast. I don't think anyone would complain about what happened with this suppression gene drive. However, if the situation is just a little bit different, things can change. For example, if your drive efficiency is a little lower, still enough to eliminate a simple population, just not quite as good. Or if we're working in a difficult organism, like haplodiploids, such as these invasive fire ants, or maybe the organism just has a slightly lower level of dispersal compared to what we saw on the previous slide. Let's see what happens when we release this suppression drive. So we see initially it's doing a good job, but wait, something kind of weird is happening now. Some of those blue wild-type individuals manage to escape the drive and move into these empty areas. In the empty areas, there's much less competition, so they can have a lot more offspring than normal. The red gene drive is still present. It's constantly chasing after these wild-type individuals, but it never quite manages to catch up. And these conditions can persist for a very, very long time, sometimes indefinitely. a gene drive that does this, of course, is a failure in some ways. It made the population quite a lot lower than the starting level. So maybe it's doing some good, but it definitely didn't get rid of the whole population. And the longer this chasing goes on, the more wild-type alleles we need to convert to drive alleles. If your gene drive was normally good enough to avoid functional resistance in a panmictic population, it might not be good enough in a spatial model. And the moment we get functional resistance, it's game over for the drive. So this means that we need much higher performance gene drive if we wanted to succeed in spatially explicit models due to that chasing phenomenon. And in fact, when we modeled actual gene drives, their names are at the bottom there, in a mosquito model, we found that the situation is even worse. In almost 100% of the time, there in yellow, we would get long-term chasing for, often hundreds or even thousands of weeks by the time we reach the end of the simulation. Only those two drives on the right had any significant chance of eliminating the population. And they're both hypothetical drives that involve a rearrangement of existing Anopheles gambii components. So this made us very depressed. We thought, is population suppression ever going to work in these spatial models? But then we realized we might actually have a little bit of help. Real-world ecologies are not just one species. There are often multiple interacting species. After all, there's thousands of species of mosquitoes on the planet, and only maybe a dozen or so that actually spread disease. So here, we're going to model the effect of a competing mosquito species that is not the target of our gene drive. Here's some heat maps showing all the different possible outcomes. On the top right there, you have the long-term chasing outcome. On the vertical axis is the competition factor. That's showing us how intensely are the two species competing. When that's very low, you can see that we get almost all long-term chasing outcomes. The drive is still a failure. But if we get up to around 0.4 or so, you can see that we get more successful suppression outcomes. Maybe there needs to be a little bit of chasing before successful suppression, but it still makes it possible again. So the gene drive and the competing species can work together to eliminate the target species. We don't necessarily have to make the target species so sterile that it can collapse in a laboratory environment. We just need to make it a less effective species than the competitor, even though it would normally have its own ecological niche. By reducing reproduction, it's not strong enough to keep that niche. Of course, we didn't just get more suppression outcomes when we had high competition. We also got drive loss outcomes, where the drive would just be randomly lost from the population before the population was eliminated. So it's not quite all sunshine, but that's not quite as bad of an outcome as you might think. If the drive is lost, we can simply re-release a few drive individuals and try again until we get suppression. Because it's a very random outcome, chances are eventually we'll get success. Ultimately, though, it's the malaria that we're very interested in. This can especially be true if the competing species turned out to be a much worse malaria vector than we thought. Maybe the target species were taking all the nice larval habitat near humans since they were specialized on humans. But if some of the other species move in, perhaps they can carry malaria, and now they're going to be much closer to humans than before. That means they might become much worse vectors. So let's focus on the malaria for now. In this new model, we've added humans. Mosquitoes will bite the humans most of the time when they reproduce. And when they do that, they can either give or receive malaria. And this means that our outcomes are now quite a lot more complicated. there on the left, you can see the mosquito population in blue and the malaria frequency in green. These oscillate because mosquitoes are usually seasonal, and then they both drop when we release the gene drive. The gene drive is the solid orange line. The gene drive increases at first, and it decreases both the mosquito population and the malaria rates a lot there on the left. But then the dashed line starts coming up. That is functional resistance. And pretty soon, that gets rid of the gene drive, and both the mosquitoes and malaria come back. The drive has completely failed. On the right, though, we have a more promising situation. You can see that the gene drive doesn't increase to 100%. Instead, it does chasing. And then eventually, we still get that dashed line, and the gene drive will fail. But during chasing, the mosquito population is kept at a low enough level that the malaria is eliminated before functional resistance forms. So that means that even though this gene drive failed, first because of chasing, and then because of functional resistance, it was still good enough to eliminate malaria in this target region. And in fact, that seems to be our most predicted outcome. That black dot there represents our most likely parameters. And in this model, we had actually so many different variable parameters, we had to use deep learning to analyze all of them at the same time. So we're only varying 2 of them, but behind that, you have variation in many, many other parameters regarding gene drive performance, mosquito ecology, as well as the malaria disease itself, transmission rates, and so forth. You can see that in a heavy malaria setting, most of the time, we're probably going to fail to actually eliminate the mosquitoes. Those are the blue outcomes on the left. But on the right heat map, that's about eliminating malaria. And you can see that we at least have a 90% chance of getting rid of the malaria with this particular gene drive. So this means that maybe we don't quite have to shoot for perfection in our suppression gene drives. As long as they're good enough, they might still be able to eliminate malaria over very wide regions. And as an extra little bonus, that chasing phenomenon, which is so bad at preventing mosquito elimination, will actually help us eliminate the malaria. So the red points here are the points that had a lot of chasing. The horizontal axis is the mosquito population. And you can see that usually when the population is high, we get a lot of malaria. That's the vertical axis. But in those red points, they're all clustered at the bottom. Even though their population size can sometimes be high, their malaria rates are low because the mosquitoes don't spend that much time in any given region. The drive always kicks them out, and that lets us break the cycle of malaria transmission. So even though chasing is very annoying in some ways, it can still give us a boost for getting rid of malaria. All right. Next up is confinement. So far, I've told you just about homing type gene drives. These copy themselves with just a teeny release, perhaps even just 1 or 2 migrants. These gene drives can then spread through the whole population. This is a map of shipping routes in the world. We're more interconnected than any time in previous history. If we release a gene drive in one patch, it's quite likely to eventually spread everywhere in the world that species exists. There might be a few small exceptions, extremely isolated islands, suppression drives in some of those isolated areas that will definitely eliminate the population before they have a chance to spread. But those are all very unusual situations. In almost any situation, including even some of those islands, the gene drive is probably going to get off the island and spread everywhere. Sometimes this might be what we want to do, especially for these really widespread problems like mosquitoes. But you can imagine for things like invasive species or agricultural pests, we only want to get rid of these species where they're causing problems. They might be a very important part of their native environment. We don't want to get rid of them there. And then there's political matters. If we can get a few countries to agree to releasing a gene drive, but there's one or two holdouts, they might not be okay with gene drives spreading over their borders. So we need some options for confined gene drive. So how does that work? Up there, we have zero threshold gene drives like the homing drive. A very small release, and pretty soon it spreads to both of those connected populations. Confined gene drives, though, have an introduction threshold. You can see that in the middle row, if we release them at a low level, they will be below their introduction threshold. And if a gene drive is below its introduction threshold, it is eliminated from the population. On the bottom row, we do a larger release. You can see that in the left population, it's spreading, but it never reaches a high level in the right population. Even though there's continuous migration, the gene drive is also being eliminated in that population because it always stays below the introduction threshold. So this means that the gene drive is essentially confined to mostly just the left population. So how do these kinds of gene drives work? There are, again, many different mechanisms, but we'll talk about the CRISPR toxin antidote drives, the most modern. These ones work a little bit like that homing rescue drive that I showed you earlier. They target a very important gene and convert it into a disruptor allele, which then gets removed from the population. The gene drive has a working copy of this gene, so it's usually okay. And the key aspect is that this gene drive does not copy itself. Here's the simplest version, the TARE drive, toxin antidote recessive embryo. It makes recessive lethal alleles by targeting a haplosufficient but essential gene. So here's how that's going to work. In the germline, we convert the wild type allele into a disrupted allele. And then we get meiosis and fertilization. And after that, we can get that maternally deposited Cas9 and guide RNA doing additional cleavage. For a TARE drive, we actually want that to happen. So that means it's a lot easier to make than a homing gene drive. The reason we want that to happen is that the whole point of this gene drive is to make disrupted alleles. Anytime we have disrupted allele homozygotes, those individuals are dead. So we convert the wild type allele to disrupted allele, and then we remove those from the population, leaving only the gene drive allele. That is how the gene drive increases in frequency. And you can see that even though the gene drive is with a disrupted allele at the bottom, it's a haplosufficient gene. One copy of the gene drive will therefore always be okay. It always has a working version of that target gene. Here's a model of the performance. Most of the time, the drive fitness will be pretty high. Not quite 1, which is wild type fitness, but pretty high. You can see then that we'll only have gene drive success, that's the blue area, if we release at around 10 to 20% introduction frequency. If we're below the dashed line in the gray area, the gene drive will be below its threshold and it will be eliminated. So that's how the TARE drive can be confined to a target population. If a TARE drive is not enough, we can use a 2-locus TARE drive, which is basically 2 TARE alleles that need each other to survive. You can see in this one, the minimum introduction threshold is 20%. That's on the vertical axis there. In this model, we've got 2 connected populations. So you can see that in the right population, if the migration rate, the horizontal axis, is high enough, we'll get a drive invasion. That might not be what we want. But if the migration is less than around 1.5%, then we're in that black area of the right heat map. That means the drive has not reached a high level in the second population. Its equilibrium is at a very, very low level, and the drive is essentially confined to the left population. So this is how we can get drive confinement even with 2 separate populations connected by migration. And from a population genetics point of view, 1 to 1.5% migration is actually quite high. So usually this 2-locus tear drive will provide enough confinement for most possible situations where a regular tear drive is not confined enough. But if not, we actually have a ton of other tear drive designs with even more confinement, just different configurations. So we were quite interested in testing that drive. You can see here that it looks just like the homing rescue drives, but instead of targeting its insertion site with the guide RNAs, it targets a little downstream so that it won't copy itself. When we released this drive into the population in blue, it was very effective and soon reached 100%. But tear drive has a big problem. It's simply not strong enough to do effective population suppression. There are other variants of toxin antidote drives that are strong enough. We've got a couple interesting spatial modeling papers for those that I'd encourage you to check out if you like the spatial modeling you saw in the last section. But this kind of drive might be difficult to make. We think we have a simpler answer to the problem of confined population suppression, and that is the tethered drive. Basically, we'll make a homing suppression drive that does not have Cas9, so it can't spread by itself. The tear drive, though, carries a good version of Cas9. So the confined drive spreads through just the target population, but not to other populations. And then we release the split homing suppression drive. It can now spread in the first population because everyone has Cas9. So it will effectively eliminate that population. But then when a few migrants try to move to a distant population, there's no Cas9 in that population. So it won't be able to spread there. The drive is essentially tethered to the confined drive. So that's how we can do confined population suppression. Oops. Here was their first attempt at making that in Anopheles-Stevens eye. We had 4 guide RNAs, and ultimately it did work. But it didn't work as well as we wanted. The way we expressed those guide RNAs worked fantastically well in fruit flies, but even though it's something that should have been cross-compatible between different species, simply didn't work that well in mosquitoes. We probably didn't have a strong enough promoter. So we still got some cleaving, but not as much as we should. You can see on the left there is a computational model based on the actual gene drive performance. If we release it at a high enough level, the drive will still be successful and spread. If we don't release it at a high level and we have some fitness costs, which we almost certainly do for this drive, then it will be eliminated. So you could say we've made a more confined version than we wanted to, if we want to think about things in an optimistic way. But we should be able to get the cut rates up quite a lot higher without too much extra effort. That's ongoing right now. All right, one more topic. There's a different type of drive that I didn't tell you about, the self-limiting drive. This drive might be able to spread at first, but over time will be eliminated. And in fact, if it's a suppression self-limiting drive, it won't even be able to spread in the first place with a single release. So here, we're not releasing this gene drive just once at the beginning. We're releasing the gene drive in that entire shaded interval every single generation. So much higher release size than homing drives, and even quite a lot higher than those confined drives. You can see that the moment we stop releasing this suppression drive in the bottom graph, it starts declining in frequency. But it still did quite a lot of population suppression before that. So this is if you need even more safety for your drive. And it's actually very similar to existing techniques. The sterile insect technique, or sterile male technique, as you might imagine, involves releasing a lot of sterile males. They mate with the females, females won't reproduce, the population goes down. But we need huge releases for this to be effective. You can see on the far left graph, red is where we successfully eliminated the population, and our release ratio has to be at least 4.3. That's if we don't have any fitness costs at all, which is an extremely idealized scenario. Usually a release ratio needs to be around 10 or 15. And by release ratio, I mean the number of males released every single generation compared to the number of males in the starting population. So that's huge. That's more mosquitoes or other organisms than is in the entire population. That's a lot more. Very inefficient. Sometimes, though, this technique works great. It eliminated the New World screwworm from all of North America. That's a nasty guy that burrows into livestock and makes infections. There have been some improvements, like RIDL. That's a genetic sterilization method. So lower fitness costs, higher efficiency. It lasts a little bit longer in the population, but ultimately still requires high release sizes. We have several different techniques using gene drive, though, that can make these release sizes a lot lower. I won't get into all the details of these, but you can see that if the drive conversion rate is high, well, then the release size is a lot lower. One of those is called RID, release of insects with dominant drive alleles. So the drive allele is dominant sterol in females, gets very quickly removed. That's why this gene drive can't increase in the whole population without repeated releases. The nice thing about this is that resistance alleles are also dominant sterol. So pretty much all the progeny of released males will be sterile, except, well, all the female progeny. The male progeny will often still receive the drive and be able to spread it more in the next generation. So you can see when we turned up our release size, even to a pretty modest level, we were able to suppress a population of a few thousand flies. We're hoping in the future, as you can see in this diagram of our newly injected Aedes aegypti mosquitoes, to make similar drives in other species. Still far more efficient than existing techniques, but much more confined than a gene drive. We hope that they can be regulated a lot like RIDL. Now back to Anopheles stevensii. So we were a little disappointed with our homing drive performance and our tear drive performance. We call the tear drive VasiCas9 here. But when we put them together, all of the females were sterile, just like your self-limiting systems. And in the progeny of the males, HSD-DSX, the suppression drive, was inherited by 100% of the progeny, a perfect gene drive. So if RID can give us 10 times more efficiency than existing methods, this could give us 100 times more efficiency, depending on exactly how you count it. So we're working on putting those together and might then have the ultimate self-limiting suppression system, hopefully in the not too distant future. All right, that's all I've got for you today. I'd like to thank my lab members who generated almost all the data that you saw here. Any questions? How difficult do you think that's going to be politically to get field studies underway? That's going to greatly vary between different countries. There's a lot of countries in Africa that are already working with very large gene drive organizations to start doing initial training and education and preparation for releases. My guess is that in those countries with their huge problems, there's going to be quite a lot of motivation to get these releases done, probably shortly after gene drives become good enough to actually release, which might be very soon. So some of these groups are hoping to do gene drive releases, possibly on islands, possibly the mainland, by the end of the decade in Anopheles gambiae. For other applications, if there's success in Anopheles gambiae, I think they might definitely get a boost. But there are definitely some places that are more eager than others. In Europe, for example, I don't think they're going to be releasing any gene drives for a while. Places like Australia and New Zealand that have an extremely large number of pets, they might be completely fine to release a gene drive that has, even reasonable levels of confinement. And then other countries might be somewhere in between. my guess is that there'll be a lot more acceptance of gene drive if the first few trials go reasonably well. Right now, though, the landscape is already changing for many genetically modified organisms. The United States uses quite a lot. There have been RIDL, self-limiting suppression system releases, and even the EU and China are changing their laws to allow certain genetic modifications in their crops. These are only modifications that theoretically could happen naturally, but it's still moving in the direction of greater acceptance. And I suspect that since most people at least are probably not planning on eating these genetically modified mosquitoes or other organisms, that it might be easier to convince people to use genetic modification to solve some of their problems rather than just giving them something to eat that they may not want to eat.
So my sense is we know a bit more about evolution of resistance to like natural selfish genetic elements. But talk about resistance arising here, like sort of in the locus that's the target of this drive. Is there anything known about like evolution of resistance to gene drives via like other organisms? I think it depends on
So
the target is.
to a large extent, yes. Natural gene drives are much more diverse than our engineered gene drives with a similar diversity of resistance mechanisms. Those, I wouldn't say they're one of the best studied areas of science, but there are a lot of interesting stories out there. For engineered gene drives, it's a little bit more of a complicated question. Most resistance we've seen is resistance that the gene drive makes at the target sequence. You can also imagine natural resistance from changed target sequences. It's one reason why we might require more gRNAs than simple models indicate. Other type of resistance is a little trickier. Actually, maybe a couple groups have kind of made their own resistance as a safety mechanism, an allele that basically completely shuts down the gene drive whenever they meet. But there could be other trans-acting effectors in genomes. In a study of genetically diverse flies, we found that certain mutations likely cause more embryo resistance to formation from that maternally deposited Cas9. So it's not quite direct resistance to a gene drive, but it's indirect and would definitely have a huge effect on the population dynamics. We saw in different genetic backgrounds, even when it was just half the genetic background that was different, the embryo resistance rates went from around 10% to 80%. So that's the difference between successful suppression and complete failure in many cases. And even for modification drives, that can have large effects. Good question. So the short answer is that the rate a gene drive advances through the population is proportional to the generation time. So that means for things like elephants, it's going to take a really long time to do gene drive, even if you had something very important, like trying to rescue them all from a new elephant plague. It'll work best in things with fast generation times, mice, rats, definitely many types of insects. But for other things, such as certain fish or certain types of ants, a generation might be a few years. In those cases, it won't directly change the dynamics of the gene drive other than making it slower. So maybe that would be a big disadvantage, or maybe gene drive is still the only way to solve the problem, in which case you might even be willing to wait a few decades, to have a complete solution. Now, if you're dealing with other ecological aspects to the organism, such as types of competition and so forth, those can also have very large effects, but that'll be different depending on exactly what those effects are. For example, if organisms make a lot of offspring that compete rather than the adults competing, that can make that chasing phenomenon worse. That's one of the reasons why our mosquito-specific model was more difficult to eliminate the population than in a kind of a simpler, more generally applicable model. Thanks, everyone.